I'm Laura Murphy-Oates, and this is The Full Story. This episode, we're looking at some of the basic questions lots of people have been asking about coronavirus. Questions like, how does the virus exist outside the body? And how long for?
I think a rule of thumb would be perhaps up to 48 to 72 hours as a way of guiding how we should undertake our public health response and cleanliness response to COVID-19.
And what's the difference between soap and hand sanitizer?
If you miss a spot, it does not matter how good your soap is, whatever good means, or how hot your water is, you miss the spot. And the virus might still hide away there. That's the key. Don't worry about the type of soap, don't worry about the temperature, and spend the time washing your hands. That's what you should be focusing on.
Today, the science behind coronavirus. It's Thursday, the 16th of April.
So, Deenan, the coronavirus, SARS-CoV-2, which causes, of course, the infection, the disease, COVID-19, is spreading from person to person. And a lot of people are really anxious about whether to touch door handles or even shake hands.
Sarah Bosley is the health editor of The Guardian in London. She spoke to Deenan Pillay, Professor of Virology at University College London, for the podcast Science Weekly, where this audio first aired.
Can you tell me, how does a virus live or survive outside of the human body?
Viruses differ from each other with regard to their chemical composition. And that, to a large extent, determines how much they can survive outside of the body, outside of a cell. Of course, all viruses are what are called obligate intracellular parasites. In other words, they have to be inside a cell, inside a body, whether that be an animal, a plant, a human, or indeed a bacteria. But they need to be inside of a cell in order to replicate. And therefore, in general, they don't survive for long periods of time outside the body. Having said that, viruses do differ by their chemical composition. SARS-CoV-2 is part of the family of coronaviruses. Coronaviruses are characterized by having around the actual genes and proteins that make it up a lipid, a fat, what's called envelope. That is needed in order for the virus to enter a cell. But that also makes it very susceptible to the sort of environments where this fat envelope will disappear. And of course, if the fat envelope disappears, the virus loses infectivity. So that is what determines how long this virus exists outside of the body.
There's been research into the other coronaviruses that are closely related, basically MERS and SARS, and how long they can survive on different surfaces. What does that research tell us?
The research tells us that this class of viruses may exist for 24, even 48 hours outside of the body. Having said that, the sort of research that can be done is to make pure concentrations of virus, put these clearly onto different surfaces. People have compared, for instance, steel bars, steel materials versus plastics versus paper, and then test how long those viruses remain infectious. Of course, in reality, what we want to do it is how transmissible in the setting that we're existing as humans, how transmissible is this virus. So it's a function of how much virus is there. It's a function of what proportion of that virus actually can replicate. And of course, then taking hold in the body, that's a function of our immune systems. So the overall infectivity is a function of a number of things, including the transmissibility of a virus particle. Clearly, compared to other coronaviruses, just from epidemiological studies, we know that this is a very transmissible virus. That's exemplified by the data coming from cruise ships, where you've got a captive population where the virus is circulating and it's a high hit rate.
And there has been some suggestion that the virus could last up to 9 days.
There are data that say that. I think we've got to remember that when these sort of experiments are done to generate the sort of data you mention, the virus is being produced in large amounts and they're placed on the surface in a way that will maximize how long they can last and the concentration is high. But I think in practice, I think a rule of thumb would be perhaps up to 48 to 72 hours as a way of guiding how we should undertake our public health response and cleanliness response to COVID-19. The important thing with regard to transmission of this virus, however, is that wherever it is on surfaces, it will only be someone touching that surface and putting their hand to their mouth, which will lead potentially to an infection, which is, of course, the underlying philosophy around the obsession that we should all have with hand washing and keeping our hands clean.
And we were talking about hard surfaces, how the virus lasts potentially quite a while on there. What about soft surfaces?
Well, things such as paper, wool. I mean, the questions come up whether scarves, for instance. I see a lot of people walking around with scarves over their mouths. We've got to remember that the virus, when it goes onto a surface, it's not just the virus. It is, of course, a virus within a droplet. So if, for instance, it's covered with proteinaceous material from sputum that come from the cells in the lung from which the virus has been coughed up, then clearly that virus will last longer on the surface than if it's without that. And as I mentioned, with this what's called lipid envelope, the fat envelope of the virus, it will quite rapidly be inactivated. So its survival on material, whatever that material, whether it's a scarf, whether it's a door handle, will also depend on what other material from the body is protecting it. So although the pure virus may survive, as the experiments show, better on hard metal surfaces, for instance, I think in practice it's far more determined by what is surrounding that virus, which will allow it to survive or not for various lengths of time.
I suppose we are a surface ourselves, aren't we? Our hands, our skin. Has there been research into how long this virus or viruses like it could live on our hands?
You know, it's a difficult set of experiments to do. I think the most important thing about hands is that we touch our faces and our mouths a hell of a lot. And it's amazing when studies have been done. And one study, for instance, took medical students who were aware that this was about seeing how long they could go without touching their faces. And it demonstrated that on average, they would touch their face or hair something like 20 to 30 times an hour.
Goodness me.
And touching their mouths, putting their fingers in their mouths on average 4 times an hour. That is in a situation where people are aware of this. You will see that becomes by far the biggest risk for then touching a virus, touching a surface with the virus and then transmitting it. This is difficult behavioural stuff.
Sometimes it's the very simple decisions that are the really hard ones. For instance, if you're standing on a train or a bus and your nose is itching, what do you do? Do you touch it with a scarf or with your hand?
I would encourage everyone to have tissues with them and to use those and throw them away safely. And of course, whatever we wipe our nose with, it is very likely that will be potentially transmissible to others if it contains virus. And that's the importance of washing our hands the whole time.
And finally, we've been asked by lots of people, lots of listeners, whether it's safe to get in a swimming pool. Obviously, the virus is transmitted by water droplets.
My answer to that is the chlorine will pretty much do away with the virus. It depends what you get up to in the changing rooms that matter.
Thank you very much, Professor Dean Pillay.
Thank you.
Next, the science of soap.
Okay, so just to start with, can you introduce yourself?
Nicola Davis is a science reporter for The Guardian.
With your name, your title and your affiliation so that we have that correct.
Yes, I'm Professor Páll Þórðarson, better known though as Palli by everyone. I'm a professor of chemistry at the School of Chemistry at the University of New South Wales in Sydney, Australia.
So, Palli, you started tweeting about why soap and water is so important in tackling the coronavirus. Why do you think it's important that we understand how that works?
I think it's important because people like to know why they're told to do certain things. And in this particular case, it may not seem very obvious to people why you can use something as simple as soap to kill a virus. Whereas we're also told at the same time, there's no drug, even if you had $1 million, there's no drug that can save you from the virus once you have it. It's only your own body that can fight it off.
And when we talk about soap killing a virus, let's talk about what we actually mean there. So what's happening when we're using soap and water?
But before I answer the question, we need to go back and clarify that our viruses are not alive. So we're not actually killing viruses, we're just making them inactive. Viruses essentially nano-assemblies of proteins, RNA, and lipids. So these three types of chemicals come together and they what we call self-assemble. You get the protein self-assembling of the RNA, and then you get this lipid coat, like a greasy film of molecules around it. Now, that turns out to be the weakest link in the structure. So once, if you have the virus on your hands, it sticks to the hand a bit because it's greasy. But if you have soapy water, the soap molecules, they actually interact with and disrupt this greasy lipid bilayer around the virus. And they start to tear it apart. And when they do that, some of the proteins that are anchored into the membrane will fall off. The connections, the weak, what we call non-covalent interactions between the proteins and the virus shell start to come apart. And the whole virus just basically falls apart like a house of cards and becomes inactive.
So, Palli, it sounds to me like what you're saying is that the substances that make up the soap basically interact with this virus kind of package and make it fall apart. And once it's fallen apart, that virus can no longer infect your cells. Is that correct?
Yes, that's correct. The soap molecules, they are very similar to the molecules that make up this greasy layer around the virus. And that's actually the key. That's why they interact so strongly and they disrupt that membrane. Once they've done that, the virus starts to fall apart. And there's no way it can self-assemble back into an active particle. Viruses can always self-assemble together inside our own cells. It's actually quite unique conditions there. And once you tear it apart in soapy water, the bits and pieces that made up the virus will just float away and you don't have to worry about it again. It's completely gone.
So when we're talking about the soap here that people might be using, is this any bar of soap, anything I've got knocking around in the bathroom, or does it have to be a particular kind?
Pretty much every soap should do. Now, I will emphasize that this hasn't actually been studied scientifically in great details, but given that soap molecules are so similar to the lipids, and soap is designed to dissolve grease and dirt, and grease and dirt is made out of, to a large degree, same sort of fats and proteins that viruses. Soap that has been designed to dissolve that will dissolve the viruses. And it doesn't even matter whether it's what we call synthetic soap, the soap molecules in those, or what some people call organic soap, the chemicals in those soaps. They will all do the job, even though it's slightly different. There's no reason to think they will be very different in terms of interacting with the lipid membrane when we're washing the virus away.
And what about the temperature of the water? Does it matter if you're using hot water or cold water? Should people go for the hottest water they can handle? What's the advice on that?
Well, that's a good question, because from a sort of chemical intuition point of view, you might think hotter water might be better. But I've seen several people who are experts on infection control argue that studies indicate it doesn't actually matter. And I think that comes to a different point, which is just like your question previously, what kind of soap? It's really not about how, can we say, active the soap is or how hot the water is. It is how you apply it on your hands. If you apply it well on your hands, and this is the 20-second advice, you will not miss a spot. If you miss a spot, it does not matter how good your soap is, whatever good means, or how hot your water is, you miss the spot. And the virus might still hide away there. That's the key. Don't worry about the type of soap. Don't worry about the temperature, and spend the time washing your hands. That's what you should be focusing on.
And just to reiterate, I think the advice at the moment is that we should be singing Happy Birthday twice when we're washing our hands. Or do you have an alternative song that you prefer?
I really need to start to find a song. I would not do Happy Birthday. I'll find something from Rage Against the Machine or whatever. But yeah, just make it long enough.
So I wanted to go back to something you just said, which is that there hasn't been an awful lot of research in how cleaning products basically deactivate these viruses. Why do you think that is? And what do you think needs to happen?
Well, infection control in general has been relatively neglected. And we are paying the price for that really big time now. We thankfully did have some studies, especially focusing on influenza, that did show that hand wash with soap is efficient in getting rid of influenza viruses. And influenza viruses are very similar in terms of vulnerability to soap and alcohol. But we haven't studied these things in details. Does it really matter? I don't think it should matter which kind of soap, what kind of soap you use, or temperature. But these things haven't been studied, and simply because the scientific and medical research communities have neglected, to a large degree, infectious diseases, including viruses. And we are paying the price for that today, unfortunately.
Let's move on to the question of hand sanitizers. So we've been told that if you can't wash your hands with soap and water, using a hand sanitizer will also help in the fight against the coronavirus. Hand sanitizer works slightly differently. Can you explain how that helps to deactivate the coronavirus?
Yes. What happens with alcohol and proteins in particular is that the very nature of the interaction and hold the protein structure together and hold them in the right fold changes when you change from water to alcohol. So those interactions all become weak and much weaker. And essentially the proteins on the surface of the virus and probably the lipid bilayer itself just crumbles down. And that's how alcohol-based proteins probably mainly deactivate viruses. And they are very, very effective as long as you have the right concentration. It needs to be more than 60-70%. And again, the only downside is you need to make sure you don't miss any spots on your hand.
And lastly, keeping our hands clean is one thing, but something a lot of our listeners have been contacting us about is objects. Listeners Simon and Mark, for example, asked about things in a supermarket. And another listener, Rob, was wondering whether he could become infected with the coronavirus through handling his daily Guardian newspaper. What's your advice here?
With treatment of surfaces, the general advice for hard surfaces like steel and so on is any kind of antibacterial cleaning products that, for instance, contain bleach or chlorine or hydrogen peroxide will do the job. If you can't use them or can't find them, in this particular case, soap probably should do, or some sort of detergents, as long as you wrap it well in. Now, I don't know about newspapers. You don't want to clean them with soap or bleach. But I think it's a little bit unlikely that the virus would hang around long on those sort of surfaces. And one thing about objects that sit outside and whatnot, they are probably safer in general. The virus is not thought to survive long in sunlight, possibly even less than an hour. So if the newspaper was on a newspaper stand or was lying out on the pavement, it should be fine. But if it comes with a plastic coating, you should wash your hands before, rip it off, and wash your hands again. That's probably the best advice I could give.
Pali, thanks so much for joining us. It's been great having you on the show.
Thank you. It's my pleasure.
That's it for today. This episode first aired on the Science Weekly podcast, made by the Guardian team in London. We will be featuring more of their episodes in our feed. And if you like these episodes, check out Science Weekly on the Guardian website or subscribe by searching for Science Weekly on whatever app you get your podcasts on. Thanks to Professor Pal Thordarson and Professor Dean Pillay for their time. You can find more reporting from Sarah and Nicola at theguardian.com. This episode was produced by India Rakison, Max Sanderson, David Waters, Danielle Stevens, and Joe Koning. Okay, catch you tomorrow.